All right, so today we're going to be making nutrient agar plates. In Biology 439, students must do an independent research project, and so a student has requested nutrient agar plates for their project. So we're going to go ahead and make those today. Now, we're only going to be making 250 milliliters, so we're going to be using this 500 milliliter Erlenmeyer flask to do that. So now my balance is on. I'm going to put the weigh boat on and zero it out. Clean spatula, and we're going to measure out the 2 grams of nutrient broth powder. Now I need to add the agar, which will then solidify, so that way the student is able to plate bacteria and get it to form colonies, which means we'll need about 5 grams of this agar. So grab another spatula, and then we'll weigh out our 5 grams. Okay, so we're good with that. All right, so in this cylinder I have 250 milliliters of water. I'm going to pour that in. And we need to get this nutrient broth powder to dissolve before we put it in the autoclave. So I've got a stir bar here. I'm going to drop that in. I'm going to add in my nutrient broth powder. Turn on the stir plate here. Let that stir. It doesn't need to stir for very long. And what's in this is a combination of yeast extract and meat extract. Essentially, it has ground up dehydrated yeast cells that have sugars, proteins, amino acids, fats, vitamins, everything that a microorganism would need to grow. Okay? So now what I'm going to do is I'm going to add in my agar and let that mix. And that should be enough mixing. So now I need to retrieve my stir bar. Now what I'm going to do is to prepare my flask for the autoclave, I'm going to use a piece of aluminum foil. And what I like to do is I like to do this and then put a stir bar in the foil, because when this agar dissolves in the autoclave, it sinks to the bottom. We want it to have it evenly mixed. And it's easy to do that by just taking a stir bar, putting it in the aluminum foil, creating a little butterfly, and then landing that butterfly on the top, and crimping it on the flask. What happens here is when we take this out, we won't have any airborne contaminants that can get inside of our flask. When the flask comes out of the autoclave, we'll just peel the foil back, and that stir bar will drop down right in there. We put it on the stir plate and let it stir, and everything will evenly distribute before we pour it into sterile petri dishes. So in the autoclave, we just put a little piece of tape on this, put it in the autoclave. It will autoclave for about 1 hour, and then it'll be ready to cool in a water bath and then pour it into petri dishes.
1 hour later.
All right, so our flask has come out of the autoclave. We put it in this 60°C water bath for it to cool down. It is now ready to pour. So I'm just going to bring this over here. So this just needs a minute or so just to stir. Now I'm going to be doing this, I have to do this aseptically. So the bench has already been cleaned with a disinfectant. We don't want to introduce any airborne contaminants into our plates. These petri dishes are sterile. Now there is a potential that when I pour the agar out of the flask in these dishes, I could introduce contamination that may be on the outside surface of this. So the agar is stirred enough now. What I'm going to do is I'm going to take my little foil cap off and then run this opening through the flame of the Bunsen burner. That decontaminates that opening so that we remove any airborne contaminants. What I like to do is I like to have my petri dishes on the edge of the bench, have the flask at the same level. I take a lid off of one and I pour this so that I just fill the bottom and then stop. That's about 25 milliliters of the melted nutrient agar. So 1, 2, 3, 4, and our last dish. And we want to make sure we pour slow enough that we don't lose our stir bar into that last plate. Put that right there. Now this flask will get flushed out to remove any residual agar. The plates, we just push them away onto the bench, and then we are finished pouring these plates. These plates will be ready to use in about 24 hours, so a student can have them for their project.